One of the most common ways to use the knowledge portals is to check whether there is human genetic evidence that supports the potential role of a gene of interest in a trait or disease of interest. Here we'll illustrate the genetic support workflow that can help you do that. We'll use the Type 2 Diabetes Knowledge Portal for this example, but this workflow is applicable to most other knowledge portals. This video will give you a high-level overview, and you can find all the details under the Help menu. In this example, we'll check whether there's evidence for the role of the SYN3A gene in type 2 diabetes. Starting from the home page, start typing a gene name, find the correct one, and select Search Region. This takes you to the region page, showing genetic associations across all phenotypes in the portal, in the region spanning the gene and its flanking sequences. Check the phenome-wide association, or PheWAS plot, near the top of the page, showing genome-wide significant associations across the region. If there's human genetic support for the role of a gene in a phenotype, there will usually be at least one genome-wide significant variant association for that phenotype on the region page. The dotted line indicates genome-wide significance, so you'll want to look for points above it. We see that the type 2 diabetes association is above this line. Next, let's look at the gene page. To get there, click the appropriate blue box at the top of the region page. First, let's check the gene-level association scores for SYN3A that are calculated from common variant associations. Click the Common Variant Associations tab to see these. The gene-level association scores shown on this PheWAS plot are calculated using the MAGMA algorithm. A generally accepted significance score for MAGMA results is p-value of less than 2.5 times 10 to the negative sixth, as indicated by the dotted line. So we can see that the SYN3A association for type 2 diabetes is above this line. However, we should remember that a MAGMA significant result indicates that the gene is close to a significantly associated variant and doesn't necessarily mean that the gene itself is causal for the phenotype. Next, we'll click the Rare Variant Associations tab to check gene-level scores for SYN3A calculated from rare variant associations from exome-wide analysis. The upper dotted line indicates exome-wide gene-level significance, and the lower dotted line indicates nominal significance. We see that SYN3A doesn't meet nominal significance for T2D. However, it does have a nominally significant association for a related phenotype, random glucose. To see a simple summary of the genetic evidence, check the Human Genetic Evidence Calculator score. This is a tool that takes into account both common variant and rare variant associations to calculate a score representing the strength of genetic evidence for the gene. Here we see that despite the lack of strong rare variant evidence for the involvement of SYN3A in T2D, the overall evidence is very strong. Finally, the knowledge portals include expert-curated lists of predicted effector genes for many diseases and traits. Scroll to the bottom of the gene page to see the lists on which your gene appears. We see that SYN3A is included on 4 different lists of predicted effector genes for type 2 diabetes, and also appears on lists for some other traits. Click the View button to navigate to a list and see all of the evidence supporting that prediction. We're happy to help you evaluate the evidence in the knowledge portals. Please contact us.